Hello, everyone. Welcome to today's live broadcast, Replacing Settle Plates with Active Air Sampling. I'm Felisa Mirsal, the Science Editor of Pharmaceutical Technology, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by Pharmaceutical Technology and sponsored by Particle Measuring Systems. I would like to share a statement from our sponsor. Particle Measuring Systems provides the tools and industry experts to detect, analyze, and manage cleanroom contamination. It is the only company that provides complete contamination monitoring services, including remote and mobile contamination monitors, microbial and particle contamination monitors, environmental monitoring systems, data management, including data integrity, education, and advisory services for risk assessments, project management, and GMP. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark Help widget in the dock at the bottom of your presentation window. I would now like to introduce today's speakers. We are pleased to be joined today by Dr. Frank Panafin and Heather Mason. Dr. Panafin has wide-ranging experience in the field of applied pharmaceutical microbiology and serves as the Business Development Creator, EMEA, at Particle Measuring Systems. Frank has a diploma in chemistry from the University of Bielefeld and a doctorate in molecular and cell biology from the University of Osnabrück. Heather Mason is a microbial specialist working with the Americas sales managers at Particle Measuring Systems, providing insight and customer assistance with microbial monitoring process and products. Thank you for joining us today. Dr. Panafin, please get us started.
Thanks a lot, Felisa. Thanks a lot for the people attending this call. I'm really happy to present you today our concept of how to replace settle plates with active air sampling. Would like to give you an indication that this material has been gathered by three people of our organization, Paola Lazzeri, Anna Campanella, and Gilberto Dalmaso, who unfortunately could not be available to present their own data, but I wanted to give the courtesy to these people because they have been doing the work and I'm going to present today. So the purpose of this call is split into two parts. First of all, I'm going to give you some indications around the scientific background and the literature behind settle plates and the functionalities of settle plates and active air sampling. And then in the second part, I will be handling how to use a QRAM approach in terms of replacing the settle plates by active air sampling. As said, the scientific literature at the beginning will give you an understanding of the origins of the guidelines that we are typically following in the pharmaceutical industry. There is as well a part that tells you about how airborne concentrations are related to settle plate counts and how settle plates and active air samplers are managed in terms of physical efficiency. In the end, in the QRAM approach side, we are going to provide a comparison of these two technologies. And then afterwards, we are looking into how they are accepted by the auditors and finally, how to validate the replacement of the settle plates. Before I really start with the content, I would like to get some questions to you, and Felisa, I can hand over to you.
Thank you, Dr. Panafin. I'd like to ask our audience to participate in a brief polling question. Please click directly on the screen to enter your answer. Here is the question. Do you use settle plates or passive air monitoring in critical environments at your company? Yes or no? Thank you for participating in our poll. I'd like to ask our audience another polling question. Again, please click directly on the screen to enter your answer. Here is the question. What is the main reason to use settle plates in critical areas? GMP requirements, important monitoring data. We do not use settle plates. Thank you for participating in our poll.
Thank you, Felisa, for taking these questions. So, as I promised, we will now go into the literature of today's talk and where does all this come from. So, originally, and this is really surprising, but in 1963, the first federal standard 209 defined the clean room air cleanliness classification. So that was a standard that was talking about particles around 0.5 micron and how they are expected in terms of cubic feet contamination. And this is directly linked to a study that Noble, Littwitz, and Kingston published regarding the size distribution of airborne particles and microorganisms. In this respect, you need to understand that this was done in healthcare facilities, so mainly in hospitals and similar settings. We need to understand that the medium equivalent particle diameter was greater than 10 microns. This is an important thing to know because we later come to the point of particle deposition, and 10 microns is an important information to know. Another aspect is that these people learned and understood that fungi appear to be present in the air as single spores, mainly when they are in the environment. We all know that we are in a very bad shape, whether we have in clean rooms in our environment, fungi present. So both of these studies from White's are mentioned in literature and in the GMP pharmacopoeias. Additionally, there were the EU guidelines on saddle plate counts. When they were derived from a PDA paper based on the publication from 1986 from White. So White is one of the biggest originator of data of these regulations. But please, you need to understand that in the 2000s, so 15 years later around, there were some interesting studies published that reassessed the relationship of airborne concentration and saddle plates and even asked to change the approach. So what do we know? Particle deposition, and we are testing with saddle plate the particle deposition normally. The velocity of the particle deposition is a very important parameter to define the saddle plate counts. In 2016, so not yet just some years ago only, White and Eaton, so again the same publisher, pointed out that the EU GMP maximum concentration should be revised to provide more accurate saddle plate count. So already in 2016, we learned that the old regulations or the old status of the regulation should be revised. And there were similar declarations by other authors referring to different classified environments. And therefore, we need to understand that still, at the moment, the standard value of particle deposition velocities is set to the 1986 publication of 0.46 centimeter per second. And they're still mentioned there, but originally the people that have published this data already revised the data that is considered. So therefore, it would be really great to understand some new aspects from your side, and I would like to hand over again to Felisa for some questions.
Thank you, Dr. Panafin. I'd now like to ask our audience to participate in a polling question. Please click directly on the screen to enter your answer. Here is the question. Do you test for particle deposition in your critical areas? Yes or no? Thank you for participating in this poll. Now I'd like to ask our audience to participate in another polling question. Again, please click directly on the screen to enter your answer. Here is the question. Do you plan to integrate particle deposition tests in the future? Yes or no? Thank you for participating in our poll.
Thanks a lot, Felisa, for these questions that are quite relevant. In order to understand how studies are done on particle deposition tests, we need to understand what are the requirements and what are the methodologies around it. And of course, the particles that are used in these kind of tests are usually in a sort of standard. Although in nature and in our clean rooms, we will never find those kind of standard particles, we must do, for experimental design purposes, make some certain assumptions. First of all, the particles are spherical, they are uncharged, they have a certain density, and all the deposition velocity is only done by gravity. So in that respect, the environmental standard set conditions are set to that there is no airflow perturbation and there is a uniform temperature of 25 degrees in order to reduce the Brownian molecular interference. So looking at the results from these experiments, you see that in 2000, there was a very detailed study published to evaluate the velocities of varying particle sizes in terms of sedimentation on surfaces. So when we look into a clean room that was classified according to ISO 14644, the deposition velocity of the cumulative counts of particle size can be calculated. And what you see here is that the velocity is increasing dramatically when you go above the 10 micrometers. And of course, it is depending on the size and weight of the particles. In this respect, we need to understand still, remember, the standard deposition velocity in EU GMP is set to 0.46 centimeter per second, or similarly in the FDA GMP. And when you look in a bit further into the so-called MCPs, so the microbial carrying particles, there was in the publication from 2016 from White and Eaton, you see a very similar aspect that the larger the particles, the higher deposition velocity. And of course, what you see is that in the end, deposition velocity increased as the cleanliness of the clean room increased. So again, just think about it and do remember that 0.46 centimeter per second is the standard deposition velocity and how this is reflected. If you see this, you will understand that you need a very high deposition velocity and a high concentration of MCPs in a clean room to reach these 0.46 centimeter per second. Taking this into account, we should go and visit a new standard that is just about to be released. It is the ISO 1464417, which is still in the draft shape, and I do believe in 2020 we will see the publication becoming ready. In the end, we know that particles smaller than 5 micron are most likely to be removed by the ventilation system. So this is the target of our pharmaceutical environment, and we understand that all the microorganisms and all the particles less than 5 micrometers should be removed definitely by the ventilation system, simply by the airflow and by the air exchanged. Above the 10 micrometer, we need to understand that still more than 50% will be removed from the air by surface deposition. So in the end, as a reference, think about what do we have in GMP as alert or maximum limit. The maximum limit are 20 particles per cubic centimeter. That means 20 particles greater than 10 microns are expected. So here we see immediately that there is a discrepancy around the theory of particle deposition and our GMP regulations. Emphasize again looking at this graph that was published from Agricola and presented during the ISSC in 2018. So as mentioned, particles less than 5 micrometers are removed by the ventilation system. You will not even find them in clean rooms of Class A. Particles greater than 5 are largely removed by the clean room ventilation system without settling. That means when you look into the graph, you can easily understand that 50 to 80% of the greater 5 micron particles are removed by the ventilation system. And remember, the median of human particles and the median of particles that are microbial carrying particles is 10 microns, and the average shape of a skin flake is typically thin, like 1 to 3 micrometer thin and 10 to 25 micrometer in diameter. That means these particles are the ones of interest that could be considered as risk factors in pharma. So how could we understand and how could we consider that a microbe on a large particle originating from an internal equipment will really settle on a specific small surface in Class A critical area? So how could we consider that under these conditions, microorganisms sitting on large particles of a medium diameter of 10 to 50 micrometers will really settle on a settle plate? It's really almost unlikely that we will understand the reality by using settle plates. So let me change a bit to the active air samplers. Viable air samplers have a certain efficiency, and there is a standard here. The standard is ISO 14698 from 2003. This was a standard that already quite modern, I must really say. The ISO 14698 had all aspects of risk assessment already included in the year 2003, when in our world of the pharma, it was not even considered in our regulations. Nonetheless, two factors will determine whether an air sampler is really efficient or not. It's the physical efficiency and the biological efficiency. The physical efficiency tells you how good the sampler can collect various sizes of particles, and the biological efficiency will tell you how good it can collect microbial carrying particles, meaning MCPs. How does that work? Air samplers typically accelerating the air through a nozzle. That means the air has a certain speed in the range typically of 10 to 30 meters per second. That is quite fast. With this high speed of air, particles are accelerated that are included in the airstream. And like a car that is driving too fast into a curve, the particles will not follow the airstream that is diverted on the surface of the impaction plane, which would be normally an agar. So the particles then would impact into the agar surface depending on the speed of the air, of course. How does that work with settle plates? Simply, we believe and we hope that airborne particles fall onto the agar surface just by gravity. Of course, larger particles have a higher tendency to settle on smaller ones, but we must understand that the air turbulences and the airspeed increases the suspension time of airborne particles. That means under high speed and under high air turbulences, you will have difficulties to have microorganisms or MCPs settle on surfaces like a settle plate. So what guidelines are existing about a settle plate's efficiency? If you look into the ISO 14698, it is clear that the settle plates are not considered a measure for air contamination, but they are a measure for surface contamination because they are measuring the settling of microorganisms on the surface. So they are not measuring the total number of microorganisms present in the air. They are measuring how microorganisms or particles would be settling on surfaces. So the same is mentioned in a very new actual draft of the European side. So that Europe has developed its own new standard out of the 14698. Generally speaking, the ISO 14698 should have been updated, and finally it happened that the ISO committee was not able to get aligned, so the Europeans themselves have developed a new standard that is a European standard 17141, which is in a draft stage, but in the final draft stage, and it says that settle plates at the moment are a measure of the number of microorganisms that are deposited per unit per area. It provides a more direct means of measuring potential product contamination than the measurement of the concentration of microorganisms in the air. That's true. But is that really true and is it really valid for a super clean environment with high turbulences? We just learned that especially when we have high airspeed, there is a cleaning effect from the airspeed that will, in the end, remove these MCPs from the airs and will prevent them from settling. Let us have a small look as well on how the regulations and the standards give us education and support in how to select air sampling devices. ISO 14698, they give us that the selection of contamination air sampling devices should be done upon the purpose for which the sample is required. So they tell us already now that you need to consider the data that is generated with this and what you want to do with this data, and not only sample just for the purpose of sampling. And ISO EN 17141, it tells us that the sampling method must be appropriate for the clean controlled environment to be monitored. So you need to select carefully sampling method according to what you're expecting. considering volumetric air samples, it says you need to select a suitable device with the right collection efficiency that is validated, and of course all the instrumentation that you're using must be periodically calibrated. So that's not new, but it is now written explicitly in this draft. That's why I like this new draft very nicely. So as a conclusion of the first part, we need to understand that the scientific literature and the norms and the standards that we have been looking at, it gives evidence of strengths and limits of settle plates in viable air monitoring. We understand that there is clear limits of settle plates, and from our practical experience in pharma, we know as well that the problem of settle plates is that they are easily contaminated by the operator. So now the question is, how can we use a quality approach in understanding the existing microbiological monitoring methods? How can we understand when we can use settle plates and how can we replace them by active samplers and how to implement that? This is the next question I'm going to cover. As I told you, we're looking now at a comparison of settle plates under a QRM approach, the acceptance by auditors on this, and on the other hand, how to validate a change. So let's first have a look at the comparison. We have 5 factors that we should be considering. The first one is to measure the concentration of microorganisms in the air. We need to understand that we want to know what quality the air of our surrounding manufacturing is. So in that respect, we should think about the active air samplers, measure the microorganisms in the air in a given time and a given volume. This is very good. This is what we really need, because then we can determine the quality of air. Settle plates, unfortunately, just give us the number of microorganisms that will deposit on a given area. That means it is a measure of surface cleanliness and not a measure of air cleanliness. By that meaning, it is the situation that the settle plates are not really giving quantitative but only semi-quantitative results, because you cannot give a real reference, only a reference volume and a reference surface. In active air samples, you can give that per volume and time. That's why it is a clear quantitative method. When you look into the situation whether it can detect low concentration of microorganisms under unidirectional airflow, so UAF, this is the abbreviation down, we have the issue in settle plates that the unidirectional flow causes a surface cleaning effect, and this means that the unidirectional flow will wash away the particles and settling is very much reduced in critical areas. On the other hand, an active air sampler can aspire actively air and therefore actively collect out of the environment the airborne microorganisms. Another important aspect is that you may have to investigate excursions out of specifications for the particle monitoring side of things. So just for the particle testing that you're doing in continuous and critical areas. If you have excursions there, settle plates are really not useful because there is no correlation. It uses completely a different methodology, but active air sampling as well a volumetric methodology that is aspiring particles according to their size, they are very similar in terms of methodology and can be taken into account. Last point that is really, I would say, the most important point after all, is the fact that validation of settle plates is almost impossible. The only thing you can do is a growth promotion test that shows that microorganism would have grown on the surface. Alternatively, active air samples have been validated and can be validated according to ISO 14698 with the physical and biological efficiency. So you know that microbial air samples, active air samples, are working in the right way. This is what you are not sure of when you're using settle plates, and data out of these settle plates are very questionable. Therefore, I would like now to ask you some more questions, and I would like to hand for this new interaction to Felisa, getting some new questions.
Thank you, Dr. Panafin. I'd now like to ask our audience to participate in a polling question. Please click directly on the screen to enter your answer. Here is the question. Do you have experience with false positive results in critical areas due to operator mishandling? Yes or no? Thank you for participating in this poll. Once again, I'd like to ask our audience to participate in a polling question. Please click directly on the screen to enter your answer. The question is, how often does that seem to occur? Never, 1 per year, less than 5 per year, less than 10 per year, more than 10 per year, or not applicable. Thank you for participating in our poll.
So there is a question. Why are settle plates still used in grade A? So knowing now all the scientific difficulties, the question is really why these settle plates do still have a value, or don't they have a value at all? So in the past, the traditional methods available were the only ones that allowed monitoring over long periods of production. So these methodologies were the only way to monitor for a longer time. So that was considered as one of the only reasons to use it. They were still somehow quantitative and, of course, very cheap. In reality, regulatory bodies still are accepting these methodologies and sometimes even request them. So these are the main reasons why settle plates are still used in clean rooms and in critical environments. So why do we really need to change now? And what is the reason why people are now considering to change? The first understanding is the science behind the settling. It has become more and more evident. So we know better around what happens in the unidirectional airflow, and that all the settle plates are not a measure of air quality but of surface quality. So gravitation plays a role. The cleaning effect on the unidirectional flow is important. And of course, the defined parameters are set according to the ideal conditions, so we don't really know what happens under the conditions of use. And then the regulatory point of view is that the EU GMP Annex draft from 2017, which we hope will be finally released in 2020, gave a clear indication that we should use now scientific approaches and scientific understanding based on risk-based approaches under quality risk management approach to really review the data and the methodologies you're using for each sampling point. And this requires as well an update on the systems, and every one of you may have heard that the EU regulatory bodies now request to have a contamination control strategy, which is really a new complete system that they are expecting in order to avoid contaminations in a clean room. And this is just to prove these new consequences. Risk assessment should be used to justify alternative approaches to those specified in this annex, only if these alternative approaches meet or surpass the intent of this annex. So you understand that if you want to change things and if you need to change things, understanding the science behind it, you are supposed to use quality risk management approaches to change your methodologies. So how would we normally consider then to do that effectively? What are the steps and what are the factors to introduce that into our environment? The only option to do that in a quality risk management approach is to do that in a step-by-step approach. And I will go through all of these steps now in the next slides. So, of course, you need to define at the beginning the QRM and contamination control strategy. Then you need to look into the different air samplers and choose the right sampling times and sampling conditions. And then, of course, understand the culture media that you're using. The risk assessment for monitoring especially must only assess the single sampling point location, but as well must evaluate that the sampling methods are able to capture the contamination. So we need to understand whether it is technically possible to capture the contamination. And you need to provide a rationale to prove that air continuous monitoring meets or surpasses the traditional method. In the contamination control strategy, you need to describe, therefore, as well, what are the particle and microbiological impact on your decision. when you're looking into selecting air samples, important is to use the concepts of ISO 14698, especially biological and physical efficiency, of course, and then, of course, looking to the chapter A3.2, where it is described how to select a sampling device. Alternatively, you could look in the upcoming European draft 7141, which will help you as well in finding the right air sample for your need. The sampling time and sampling rate is really important. By this, you need to take into account from a manufacturing perspective the total duration of your whole process. Let's consider you have an 8-hour process from setup to the end. And of course, you need to understand how often you're doing interventions, and especially you need to decrease the monitoring interventions as much as possible. So from your side, the interventions generally should be reduced, but especially as well the interventions for monitoring purposes, because they are unintentionally and are not necessarily needed. And then the sampling rate, when you consider the sampling rate, the whole process, so the continuity of the process need to be covered. This means you should no longer think about putting snapshots of your process by sampling microbiological samples. No, you should know what has happened throughout the whole process and not only single events. And it is absolutely clear that these measurements should not interfere with the viability of the microorganisms and should not pose any risk to the process at all. That's clear. And of course, at the end of it, that's for microbiology standpoint quite useful. You're looking typically at the culture media that is used. So you do typically a growth promotion test before use. And during the validation in a QRM approach, it is recommended as well to check whether the growth promotion still works after use. So if you would be sampling for 10 minutes, 20, 100 minutes, even up to 2 hours, after this time you need to understand whether the microorganisms would really grow and that this time would have not affected the fertility of the media. And of course, you need to correlate all the data according to the science behind it. As a conclusion, from a scientific and literature perspective, we need to understand that settle plates in unidirectional airflows and in critical environments are not really a favorable methodology. We know that the use of settle plates is very useful where there is turbulences, less airflow, etc. So still in grade C and D, we have very valuable data telling us what's happening in these gowning areas, for example. But in critical areas, the data generated by settle plates is really questionable and not validatable, and especially it is not actionable data because you simply cannot trust what is coming out. As a consequence, people will be considering at this moment of time, now that we have a new area of regulation and understanding of pharmaceutical manufacturing, to implement active air sampling by replacing settle plates by active air sampling. There are certain ways of doing this, and under these aspects, I would like to hand over now to my colleague, Heather Mason, who would be explaining to you our concept and our strategy of the company, and as well how this ties into the replacement of settle plates by active air sampling. Heather, thank you.
Thank you very much, Frank. I'm Heather Mason, Field Application Specialist at Particle Measuring Systems. So before we start the Q&A, I'd like to briefly discuss our complete sterility assurance solution. The slide on your screen shows our contamination monitors, services, training and education, and our environmental monitoring systems. So let's go into each one of those in a little bit more detail. For contamination monitors, we offer portable and remote particle and microbial instruments. We also offer a BioCat single-use impactor for the direct replacement of settle plates. The BioCat single-use complies with ISO 14698 Part 1. The culture media is validated to be stored at either room temperature or under refrigeration with a shelf life of up to 12 months. We offer this in TSA and SDA, and we offer a variety of neutralizers. These can replace settle plates in clean rooms with the ability to be used for up to 2 hours at 25 liters per minute. Some of the benefits of BioCap single use. They help to eliminate false positives by reducing the risk of contamination by improper handling of media plates. They also eliminate the time-consuming and operator-dependent cleaning and process steps associated with traditional stainless steel samplers. This includes disinfection, sanitation, sterilization, and autoclaving procedures. This can create a simplified SOP that eliminates possible injuries and handling concerns with stainless steel impactors. It reduces the risk during the introduction and withdrawal of microbiological sampling plates in sterile production areas. So next, I'd like to move on to services. So at Particle Measuring Systems, we offer advisory services. We can help you with your sample point location, risk assessment, process improvement. We do project management. We do calibration and maintenance, validation and installation, and software support. We also offer training and education. We have webinars such as today's. We have a particle college. We have a GMP academy. And we can do on-site customized training. And we have a knowledge center on particle matters. Next, our environmental monitoring systems. We have data management systems meeting 21 CFR Part 11. We do offer a Facility Pro with various configurations and software options, and we can do sampling automation. So all of this together equals your data generation from an FMS system or any of our instruments with data management, and we have a complete package of pharma integrity. Now I'd like to put this webinar back to Feliza and Frank for your question and answers. Thank you very much.
Thank you for such an informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of the presentation window. And our first question is, Dr. Panfin, you explained the physical efficiency of air monitoring devices. What about the biological efficiency?
Well, this biological efficiency can be tested by an experimental design that is described either in the ISO 14698 or in the European norm 17141. This is, on the one hand, a comparative aerosol testing, so you compare a testing with an instrument against an existing instrument that has been validated already. And then on the other hand, you can do as well a comparison against already validated monitoring devices. This is something that is described in the new European norm. It can become quite difficult to get sufficient statistical data and to get a good homogeneous distribution. So you need to be very careful on this. And therefore, it is the first one in comparative aerosol testing. It's a bit easier to do, and this is typically done by the suppliers of the air monitoring systems, so you don't need to take care about this. You just need to ask the supplier from this.
Our next question is, settle plates seem to be a measure of surface contamination. Is that correct?
Yeah, in fact, it is in reality, it is really a measure of the surface contamination rate, so how fast the surface is going to contaminate. And it is not a measure of the absolute contamination of a certain area. So as I try to explain, it is a bit odd that we try to understand the air quality by this. In essence, it is a measure of how surfaces will get contaminated over time.
Our next question is, can we do only one active air sample for 8-hour sterile fill using a BioCap, as robotic fill may not have been an option of replacing the BioCap?
Well, this is a very good question. And a robotic line, it is a really a kind of a difficulty because typically you don't have any glove entries inside, and that's why you need to have built in the a robotic way of exchanging air sampling methodologies. In this respect, it might be a chance to use a robotic arm to exchange the BioCap. We've done some studies and we're looking into that together with robotic manufacturers in the filling line environment. At least it is the best option if you only can handle one active air sampling in a line to take the BioCap, because that can sample 2 hours, so you will have at least a 2-hour overview of the 8-hour sterile fill, although I would not consider that as perfect. You need to understand whether you would be ready to capture enough contaminations by a useful risk assessment.
Next is, why do you consider settle plates as appropriate for the use in non-critical environments?
Well, in non-critical environments, the air is typically turbulent or does not even have at the same high speed like in critical areas. And under these conditions, the deposition speed of MCPs, so microbial carrier particles, is higher, and that means in this respect you will have a value that gives you better data because you do not have the cleaning effect of the airspeed. So that is why, for example, in clinical environments, so hospitals and so on, where settle plates are very often used, I see that it's quite a useful tool to understand how it is contaminated. And on the other hand, in clean rooms DNC, it could be as well a very helpful know how the environment and how the surfaces of these environments will be contaminated. So I would say it has something to do with the air exchange rate and the high speed that will of the critical environment where the settle plates may not be really useful, but in the other areas you don't have this cleaning, air cleaning efficiency, and that means it can be a helpful data.
Now, can we do sterile manufacturing in a robotic isolator without passive air sampling?
Well, I do believe yes. Anyhow, the purpose of robotic air isolators and robotic samplers are to exclude the human intervention, and therefore the most important contaminator is removed out of the environment. In that respect, I would say that removing the passive air sampling is a method to really use, let's say, to really remove false positives, because in the end, these passive air samplings, so the settle plates, will be handled in a laboratory. They need to be removed from into a lab, and these are prone to contamination by the people. And here, I must say, if you have a robotic line, you have excluded the people already. Doing afterwards a movement into a laboratory, and you don't have and you have the risk of cross contamination. I would not like to do that. Of course, it needs to be supported in the special case, of course. But I would even dare, and I would long to go to remove completely the settle plates and try to use only the active air sampling at the right points, like it is expressed in EU GMP Annex 1.
Settle plates can be used in narrow spaces, but how do you handle this with active air sampling devices?
Well, the remote sampling hats that are used, like the BioCap or the BioCap stainless steel, they are not really spaces. So these remote sampling setups can be used as well in areas where settle plates would fit. So I don't see any difficulties to use active air sampling if you use a remote sampling. Of course, in such critical areas, you should not use air sampling devices, so meaning really air sampling instruments. That should be just remote sampling setups where the instrument is outside the unit.
Next question is, passive air monitoring is used in the context of continuous monitoring for up to 4 hours. Active solutions only deliver a snapshot of the process. How do you see this aspect?
Well, as I tried to explain previously, is that active air sampling can be, and there are units available like the BioCap can be validated for more than just 10 minutes. That means if you reduce the speed of sampling and the flow of sampling and then increase the time, you can validate, for example, the BioCap is validated for 2+ hours. That means under these aspects, you can consider a continuous monitoring with active air sampling as a real alternative option. And that is why I would say, yes, it is possible to replace the continuous monitoring.
Now, unfortunately, we seem to have come to the end of our webcast. I want to thank the audience for attending and for participating in today's event. I would also like to thank our sponsor, Particle Measuring Systems, for making today's educational webcast possible. We would like to ask everyone in the audience to participate in a brief survey from our sponsor. You can see the survey to the right of your screen. You will receive an email alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you all for joining, and we will see you next time. Goodbye.